Hello, my name is Joaquin Nunes from the University of Virginia, and today I would like to give you a quick overview of the Backyard Evolution program. So first things first, I would like to thank you for signing up to our program. Our goal is to leverage your passion and your curiosity for nature to better understand the fruit fly species that live and thrive in our compost piles here in Central Virginia. We hope to learn in particular how species distributions change over time, and then eventually we hope to use that data to ask other interesting questions about ecology and genetics. An important reminder is that as part of this program, we would like you to help us collect flies. You will do this every 2 weeks from the late spring season to the early winter season. And then at the very end of the program, you will send us all of the flies that you collected. Over here, once we get your flies, we will take care to characterize the species of those flies, and we will do all sorts of cool analysis with those samples. I would like to highlight at this point the contributions that some naturalists, just like you, made last year to the program. So the Backyard Evolution program started a year ago with a collection of very passionate naturalists that helped us collect fruit flies from their compost piles. And so in this map, I am showing you all of the participants that helped us collect fruit flies. And the 4 points in red represent these 4 examples that I am now projecting on the screen. Now, at the same time that our naturalists were collecting fruit flies in their backyards, we over here in Charlottesville, we were doing fruit fly collections at a nearby orchard in Carter's Mountain. And that's going to be interesting because I hope to compare the results from your compost piles with some of these patterns that we have seen in the orchards. The general idea of these graphs that we're seeing over here is that on the x-axis, each tick represents a collection point. So the first collections were done over the summer, and then the later collections were done over the winter. So this effectively represents the passage of time and the seasons. The y-axis, on the other hand, represents the proportion that a particular species was seen at a given time at a given compost pile. And so I hope that you can see there are all sorts of really cool patterns that are emerging here in our collector sample. But I also want to highlight the number of species represented here by the different colors that were collected in these compost piles. So I want to give you some quick highlights about this species. The first species that we observe is a species that is very dear and near to our heart. This is Drosophila melanogaster, also known as the vinegar fly. And this is a very famous species of fruit flies because of its great use in a variety of studies in biology, biochemistry, and medicine. In fact, a number of Nobel Prizes have been associated with research in Drosophila melanogaster. In addition to that, we also observed in the collections Drosophila suzukiae. Now, unfortunately, Drosophila suzukiae is famous for all of the wrong reasons. This is a major pest here in the continent, and it's known to attack cherries, blueberries, raspberries, blackberries, and other fruits. In addition to that, we also observe the species Drosophila immigrans. Now, Drosophila immigrans is a type of fruit fly that is known to have relatively high cold tolerance. And this is going to be important when we go back to that graph with the different colors. We also observed Drosophila hydei, which is a known member of the compost pile community. So it's not too surprising in that sense that we observe hydei. However, a very famous bit of trivia about hydei is that this is a fruit fly that is famous for having really, really long sperm cells. So maybe in trivia night in the future, if that's a question that comes out, well, you know the answer now. And lastly, we also observe some Saprionus indianus. Saprionus indianus is known for its very pretty stripe patterns on the back of the organism. However, it's also famous for being a really recent invader. So this species is just starting to invade our continent. And so there's a lot of really interesting invasion biology that we can do to learn how this process is taking place in real time. Now, I want to go back to this figure because this figure shows some really cool patterns. For example, remember how I mentioned that Drosophila immigrans was a species of fruit fly that was known for being more tolerant to colder conditions. Well, if we look at this change of distributions on this particular panel, we notice that as the winter rolls in and temperatures become colder and colder, we do observe an increase in the amount of immigrants that our naturalists are collecting. So that sort of makes a lot of sense in terms of, like, fitting in the ecology of these organisms. The last bit that I would like to do is I would like to give you the comparison. How do these collections compare to some of these orchard collections that we've been doing here at UVA? Well, the story seems to be that in the orchards, the protagonist it's a completely different type of fly. So while in the compost file we observed that transition of fruit fly species based on their tolerance to more summer or more winter-like conditions, in the orchards the story appears to be primarily about this new invasive species, Saprionus indianus, which becomes really abundant in the fall, but then it disappears before the winter. Yet it returns, in the following year of collection. So there is a lot of really interesting and cool biological questions that we'll be able to ask with this data. But that being said, I am extremely excited to see the data that you'll collect this year. Once again, I'm so grateful for your help. And remember, if you have any questions about the program, feel free to reach out to us here at the University of Virginia. You can do so by visiting our website and looking up the different emails. Or you can also do that by scanning the QR code that is shown on the screen. With that, I want to say thank you, and I'll see you in the next video.